everyone, my name is Kristen, and today I'm going to be discussing Naegleria fowleri, which is colloquially known as the brain-eating amoeba. So, N. fowleri was first identified in 1965, and it was named after Dr. M. Fowler, the doctor and one of the first authors of this paper, where this disease and this pathogen was first identified. So N. fowleri is a eukaryote of the kingdom Protozoa, and it is a thermophilic amoeba, which means that it likes warm waters and it can be found living in lakes. And it is a cause of it causes primary amebic meningoencephalitis, or PAM for short. And PAM is a non-communicable disease because when this amoeba is spread, it goes through the nasal cavity in contaminated water, and then the amoeba makes its way up to the brain. Once it infects the brain, it can cause initial symptoms and signs like headache, fever, nausea, and vomiting, and eventually lead to confusion, seizures, hallucination, coma, and then eventually death. So this all happens in a very short time frame. At best, a patient has less than a month before death, and then at worst, it can be like less than a week. So definitely this very short time frame for diagnosis leads to a very high death rate of 97%. So since 1962, out of the 154 known infected individuals, only 4 people in the US survive. So very high death rate, but in reality, these reinfections are quite rare. Since 2012, on average, there were between 0 and 5 cases diagnosed annually in the US. So definitely it's a sporadic disease and it's not something that happens all the time. Despite this, we do see that since the disease onsets really quickly and there's not much time to diagnose and treat, it has a high death rate, which is why it is still relevant today. And so, like I mentioned before, there are only 4 survivors in the US since the 1960s. And so that means that we can clearly see what treatments these people had. And here we see the medications like antifungal and antimicrobial medications used. However, just because these 4 people survived doesn't necessarily mean that they left unscathed. Unfortunately, not all of them returned to being fully functional. And in addition, some of these medications, these very same medications used on these survivors, were used on people who unfortunately didn't make it. So these medications are definitely not bulletproof and not an 100% cure. So, therefore, in this paper published in the International Journal for Parasitology, Drugs, and Drug Resistance in December of 2021, called The Therapeutic Potential of Novel Isobenzofuranones Against Naegleria fowleri, these authors synthesized some isobenzofuranones, or compounds known to have antimicrobial and antiprotozoal effects and characteristics. They synthesized these and tested them against N. fowleri. So here are the 14 compounds that they synthesized, and of these 14, they identified 3 in particular that were especially effective against the amoeba. So here's an example of something they did where they incubated the compounds with the amoeba, and you can definitely see that there's a decrease in amoeba growing as compared to when there is control, no compound added. Some other assays they used included detection of chromatin condensation, plasma membrane permeability, generation of reactive oxygen species, and an analysis of mitochondrial function disruption. So basically all signs of cell death, cell stress, and at least hopefully inhibition of the cell's growth. So for instance, here you can see an assay they did to test for chromatin condensation, which is a hallmark of late stage cell death. So here they stained with host stain, which would stain chromatin or nucleic acids blue. And so you do see that here. And they also stained with propidium iodide, which would fluoresce as red fluorescence, which would indicate actual dead cells. So herein lies a weakness of the compounds that they use, because you do see that there is chromatin condensation. However, you would see dead cells as red, and you clearly don't see that here. So unfortunately, I don't have time to go over other experiments and figures, but this leads me to the critiques, which include that the assays did not produce very impressive results. Like we saw in the previous slides, although there does seem to be some sort of cell stress going on, the amoeba is still alive in most of these in vitro tests. So these assays themselves indicate that the compounds aren't very strong. because instead of asking for actual cell death, absence of amoeba, they're going after things like chromatin condensation, mitochondrial failure, which are more indirect. Also, the cytotoxicity tests that they did were in murine macrophages or mouse macrophages, and this might not translate to cytotoxicity in human cells, and especially in human brain cells. And then lastly, it's unknown and unclear in this paper and in my own research whether or not these isobenzofuranones can cross the blood-brain barrier, which would be a very important characteristic in medicine that is trying to target something in the brain. Which leads me to my popular article published in the Science Daily in 2018 called Cancer Drug and Antidepressants Provide Clues for Treating Brain-Eating Amoeba Infections, where they used already FDA-approved drugs to test to see if they have any effectiveness against the fowleri. So one of the perks of this was that they chose drugs that are already known to cross the blood-brain barrier, such as antidepressants like Prozac. So like this quote says, even if a drug can inhibit or kill the amoeba in a dish, it might not actually work in vivo in a real person. So that's why using these already known to work drugs to target fowleri would be very helpful. Additionally, these drugs are already FDA approved, so if they were shown to be effective against fowleri, they could be easily used in real life without having to go through the whole FDA approval process, making this a lot faster. So this is some current research on fowleri, and I hope you learned a bit. And if you have any questions or follow-up comments, please put them in the comment section. Thank you.